Welcome to Nexus Clinical. Before diving into this lecture, I want to ensure we're all on the same page. This is for general informational purposes only and does not constitute the practice of medicine, nursing, or other professional healthcare services, including the giving of medical advice. No doctor-patient or pharmacist-patient relationship is formed. Using this information and the materials linked to this content is at the user's risk. This is not intended to substitute for professional medical advice, diagnosis, or treatment. Users should not disregard or delay seeking medical advice for any medical condition they have, and they should consult their healthcare professionals for any such conditions. Clinical experts created the references, content, and clinical insight. NotebookLM, a Google AI tool, created the content, which I extensively reviewed before release. Finally, the host states that he takes all conflicts of interest seriously. Currently, there are no conflicts to disclose. For all of his disclosures and the companies he invests in or advises, he directs users to reach out independently, where he keeps an up-to-date and active list of all disclosures. This module provides a comprehensive overview of applied pharmacogenomics, focusing on the principles necessary for translating individual genetic variations into actionable clinical decisions for both small molecule drugs and advanced therapies. The molecular foundation of this discipline lies in the central dogma, which establishes the causal chain linking variations in deoxyribonucleic acid, DNA, such as single nucleotide polymorphisms, SNPs, insertions and deletions, indels, and copy number variations, CNVs, to profound differences in the function of critical drug disposition proteins, like metabolic enzymes and transporters. To manage the complexity of co-inherited variants, the field utilizes the standardized star allele nomenclature system, which translates multivariant haplotypes into a functional shorthand, diplotype, that predicts a patient's clinical phenotype, for example, poor metabolizer, ultra-rapid metabolizer. Translating raw genetic data requires proficiency in bioinformatics resources, such as DBSMP, UniProt, and the Protein Data Bank, to assess population frequencies, protein-level impact, and structural consequences of genetic changes. Beyond small molecule drugs, genetic testing has become a fundamental prerequisite for advanced therapeutic modalities. This includes germline testing for specific alleles, for example, HLA, B5701*4 Abicaver safety, tumor molecular characterization, for example, HER2 status for trastuzumab efficacy, and definitive genetic diagnoses required for gene replacement therapies like Zolgensma and Luxturna. Ultimately, the successful integration of pharmacogenomics into patient care depends on navigating the translational gauntlet, which demands the rigorous, evidence-based evaluation of tests using the ACC framework, assessing analytical validity, clinical utility, and addressing ethical, legal, and social implications. These learning objectives focus on the essential concepts and skills necessary for applying pharmacogenomics in clinical practice. First, it is important to understand the molecular foundation of this field. The central dogma of molecular biology explains how genetic variations in deoxyribonucleic acid, or DNA, establish a causal link to significant differences in the function of proteins involved in drug disposition. Students should be able to classify different types of genetic variations. These include single nucleotide polymorphisms, or SNPs, as well as insertions and deletions, known as indels, and variations in the number of copies of particular genes, called copy number variations. Predicting the likely functional consequences of these variations, such as whether they result in loss or gain of function, is also crucial. Applying nomenclature and predicting diplotypes is another key skill. This involves using the star allele system to translate complex haplotypes into a functional diplotype, which can then be used to determine a patient's metabolizer phenotype, such as poor metabolizer or ultra-rapid metabolizer. Interpreting bioinformatics data is vital as well. Utilizing resources like the Single Nucleotide Polymorphism Database, the Universal Protein Resource, and the Protein Data Bank enables analysis of genetic variants. This includes retrieving data on their population frequencies, assessing their impact on proteins, and visualizing structural changes. Understanding the relationship between genotype and clinical outcomes for enzymes is essential. For example, recognizing how poor metabolizer and ultra-rapid metabolizer phenotypes affect drug efficacy and toxicity can guide treatment decisions. Poor metabolizers may experience increased toxicity with active drugs, while ultra-rapid metabolizers may have diminished responses. Recognizing the relevance of genetic variants in drug transporters is also important. Variations in transporters, such as the solute carrier organic anion transporter family member 1B1, can lead to reduced hepatic uptake of drugs, resulting in higher systemic exposure and an increased risk of toxicity, such as statin-induced muscle problems. Identifying strategies for companion diagnostics involves understanding three main pillars. These include safety screening for adverse reactions, such as testing for the HLA-B*5701 allele before administering the drug abacavir. Target identification, exemplified by hormone receptor 2 testing for trastuzumab therapy, and resistance assessment, such as detecting KRAS mutations. Distinguishing between the rationale for different types of genetic testing is also significant. For gene replacement therapies aimed at correcting monogenic disorders, testing focuses on gene identification, such as detecting mutations in the survival motor neuron 1 gene in spinal muscular atrophy. In contrast, molecular pathology testing for chimeric antigen receptor T-cell therapies involves identifying tumor antigens like CD19. Evaluating clinical readiness involves applying a framework that considers four components: analytical validity, clinical utility, and ethical, legal, and social implications. This framework helps determine whether a pharmacogenomic test is suitable for clinical use. Finally, it is essential to address barriers to implementation. Ethical, legal, and social challenges include concerns related to genetic discrimination, data privacy, and informed consent. Recognizing and understanding these issues are critical steps toward integrating pharmacogenomics safely and effectively into healthcare practice. Pharmacogenomics involves studying how an individual's genome affects their response to drugs. It combines pharmacology principles with large-scale genomic data to support personalized medicine, aiming to improve drug effectiveness and reduce adverse effects by customizing treatments based on a person's unique genetic makeup. The core of this field is based on the central dogma of molecular biology, which explains how genetic differences can cause significant variations in drug response. The flow of genetic information from deoxyribonucleic acid, DNA, to ribonucleic acid, RNA, to protein is the core process that determines cellular function. In pharmacogenomics, this pathway is the direct causal chain linking genetic variation to clinical outcomes. A patient's genome acts as the blueprint for all proteins, including those essential for drug metabolism and action, such as metabolic enzymes, drug transporters, and drug targets. A change in a gene's DNA sequence can be transcribed into a different messenger RNA, mRNA, molecule. This, in turn, can be translated into a protein with a changed amino acid sequence, which may have decreased, increased, or no function at all. For example, a genetic variation in a cytochrome P450 (CYP) enzyme gene might produce an enzyme that metabolizes a drug much more slowly than the wild-type or common version of the enzyme. For someone taking a standard dose of a drug processed by this enzyme, this genetic difference can cause the drug to build up to toxic levels. Conversely, a variation that results in an ultra-rapidly functioning enzyme could eliminate the drug so quickly that it never reaches therapeutic levels, leading to treatment failure. Therefore, the principles of the central dogma directly explain the molecular basis of individual differences in drug response, forming the foundation of all pharmacogenomic interpretation. The human genome is remarkably similar across individuals, but it is the small percentage of variation that accounts for the diversity of human traits, including drug response. These variations, or polymorphisms, exist in several forms, each with distinct potential consequences for protein function. Single nucleotide polymorphisms are the most common type of genetic variation. They occur when a single nucleotide base, either adenine, thymine, cytosine, or guanine, is substituted for another at a specific position within the genome. These genetic variations can be categorized based on their location within the genome and their impact on the resulting protein. Synonymous or silent, Single nucleotide polymorphisms occur within protein coding regions, but do not alter the amino acid sequence of the protein. This is due to the redundancy of the genetic code, which allows different codons to code for the same amino acid. Although these types of polymorphisms were historically considered functionally neutral, emerging research indicates that some synonymous variations can influence the stability of messenger RNA or the process of splicing, subtly affecting the levels of protein expression. Non-synonymous single nucleotide polymorphisms are genetic variations that alter the amino acid sequence of a protein. These variations are further categorized into two types, missense and nonsense. Missense single nucleotide polymorphisms occur when a change in the nucleotide sequence results in a different amino acid being incorporated into the protein. The impact of this change can vary widely. It may be negligible or entirely disruptive, depending on the chemical properties of the new amino acid and its position within the protein's three-dimensional structure. For instance, a substitution occurring in a critical active site residue is more likely to impair the protein's function than one on the surface of the protein. Conversely, nonsense single nucleotide polymorphisms involve a nucleotide change that introduces a premature stop codon. This leads to the production of a truncated protein that is usually non-functional, potentially impairing the protein's overall activity. Insertions and deletions, collectively known as indels, involve the addition or removal of one or more nucleotide base pairs. Their functional impact depends heavily on their size and location within a coding region. When an indel causes a frame shift, it involves inserting or deleting a number of nucleotides that is not a multiple of 3. This type of change disrupts the triplet reading frame of the gene, scrambling the entire downstream amino acid sequence. Such alterations almost invariably lead to the formation of a premature stop codon, resulting in a severely truncated and non-functional protein. In contrast, in-frame insertions or deletions involve the addition or removal of a number of nucleotides that is a multiple of 3. This results in the inclusion or exclusion of one or more amino acids in the protein. The functional consequences of these changes depend on the importance of the affected amino acids to the structure and function of the protein. Copy number variations are large-scale structural changes in the DNA that involve either the deletion or duplication of segments of genetic material. These segments can vary greatly in size, ranging from 1 kilobase to several megabases. Such variations can include entire genes or multiple genes. When a gene is deleted completely, it results in the total loss of function for that specific gene. An individual who is homozygous for a gene deletion will not produce any protein from that gene. Conversely, gene duplications or multiplications occur when an individual carries multiple copies of a gene on a single chromosome. If these copies are functional, they can lead to a gene dose effect, which significantly increases protein expression and enzyme activity. This mechanism is primarily responsible for the phenotype observed in individuals known as ultra-rapid metabolizers for certain drug-metabolizing enzymes. The type of genetic variation present in a gene creates a predictable hierarchy of potential functional impact. A synonymous SNP is least likely to have a major effect, though subtle effects on expression are possible. The impact of a missense SNP is variable and requires careful analysis of the specific amino acid change and its structural context. In contrast, nonsense mutations, frame shift indels, and gene deletion CNVs are almost always associated with a complete loss of protein function. Conversely, gene duplication CNVs are the primary mechanism for a significant gain of function. This hierarchical framework is a critical tool for clinicians and researchers to rapidly assess the potential clinical significance of a given genetic variant. Individual SNPs rarely exist in isolation. Instead, a set of SNPs and other variations located close together on the same chromosome is often inherited as a single block. This co-inherited set of variations is known as a haplotype. Understanding haplotypes is essential because the functional effect of a gene is often determined by the combination of several variants acting in concert, rather than by a single SNP. To manage this complexity and translate it into a clinically useful format, the field of pharmacogenomics has adopted a specialized naming system known as the *** symbol allele nomenclature. This system acts as a functional shorthand, assigning a unique star allele designation to a specific haplotype that is known to confer a particular functional status. For example, the wild-type or fully functional reference allele of a gene is typically designated as *1. For instance, CYP2D6*1 represents the allele associated with normal CYP2D6 enzyme activity. Alleles defined by haplotypes containing variants that lead to altered function are assigned sequentially numbered designations. For example, *2, *3, *4, and so on. For example, the CYP2C9*3 allele is defined by a haplotype containing the missense SNP C.1075 A greater than C, which results in reduced enzyme function. Alleles representing whole gene deletions are often designated as 5, such as in CYP2D65. Alleles with multiple gene copies, duplications, are indicated with an XN suffix, such as CYP2D6*1XN, signifying multiple copies of the functional *1 allele. An individual's genetic makeup for a particular gene is described by their diplotype, which is the combination of the two star alleles inherited from each parent. For example, CYP2C9*1/*3. This diplotype is then used to predict the patient's clinical phenotype, for example, intermediate metabolizer. The star allele system is therefore more than a simple naming convention. It is a critical tool of abstraction that translates complex, multivariant haplotype data into a single, functionally annotated unit. This system provides a standardized, universal language that allows clinicians and researchers to communicate effectively about the functional consequences of genetic variation. It bridges the gap between raw DNA sequence data and clinical action, enabling a non-geneticist to make an informed therapeutic decision based on a standardized functional label. For example, the patient is a carrier of a*3 allele, without needing to interpret the specific nucleotide changes and their intricate interactions. To translate raw genetic data into clinical insight, practitioners must be proficient in using publicly available bioinformatics databases. These resources serve as comprehensive catalogs of genetic variation, protein function, and structural biology. This section provides a practical, tutorial-style guide to analyzing a clinically significant pharmacogenomic variant, using the CYP2C9*3 allele as a case study. This allele is defined by the missense SNP RS1057910, C.1075 A greater than C, which results in the amino acid substitution P.Al359Lu, I359L, and is associated with significantly reduced metabolism of numerous drugs, including the anticoagulant warfarin and the anticonvulsant phenytoin. The NCBI Database of Short Genetic Variations, DBSMP, is the primary public archive for all types of short genetic variations, including SNPs, indels, and microsatellites. It is the essential first step in characterizing a variant. Objective here is to use DBSMP to retrieve fundamental information about the RS1057910 variant, including its genomic location, molecular consequence, and population frequencies. The process begins with searching the database for the specific genetic variation. To do this, navigate to the National Center for Biotechnology Information's reference Single Nucleotide Polymorphism Database homepage and enter the reference SNP identification number, known as RSID, which in this case is RS1057910. After submitting the search, the resulting page provides a wealth of detailed information. Key fields to examine include the organism, which is Homo sapiens, and the variant type, classified as a single nucleotide variation. The alleles associated with this variant are important. The ancestral allele is A, while the variant allele is C. Some submissions also report a G allele, but in the context of the CYP2C9*3 variant, the C allele is considered clinically relevant. Further details include the precise genomic location of the variant, which is provided in terms of coordinates on the human genome reference sequence, such as on chromosome 10 using the GRCH38 assembly. The database also indicates the gene within which the variant is located. In this case, CYP2C9, and describes the functional consequence of the variation. It is classified as a missense mutation, meaning it results in a change to an amino acid in the protein product, which may impact the protein's function. The Frequencies tab is critically important because it consolidates data from large-scale sequencing projects such as the Genome Aggregation Database and the 1000 Genomes Project. This tab provides valuable insights into the distribution of the C allele, also known as the CYP2C9*3 allele, across different ethnic groups. Notably, there are significant differences in allele frequency among populations. In South Asians, the frequency is approximately 11.4%. For individuals of non-Finnish European descent, the frequency is around 6.6%. East Asian populations exhibit a lower frequency of about 3.1%, while African and African American populations have an even lower frequency, approximately 1.3%. The allele frequency data provided by DBSMP is not merely demographic trivia. It serves as a direct proxy for the pretest probability of encountering a specific variant in a patient of a given ancestry. This information has profound implications for the design and implementation of pharmacogenomic screening programs. For example, the relatively high frequency of the CYP2C9*3 allele in South Asian and European populations suggests that preemptive genotyping for this variant prior to prescribing warfarin may have a higher clinical yield and be more cost-effective in these groups compared to populations where the allele is rare, such as those of African ancestry. This allows clinicians and health systems to move from broad population-level data to an individualized assessment of risk and the potential utility of genetic testing for a specific patient. The UniProt Knowledge Base, UniProt KB, is the world's most comprehensive resource for protein sequence and functional information. It integrates data from literature and computational analyses to provide a detailed profile of a protein, including its function, structure, and associated variants. Here, the objective is to use UniProt to map the RS1057910 DNA variant to its specific protein consequence and access curated information about its functional impact. The process begins by navigating to the UniProt homepage and searching for the gene name, CYP2C9. Once the search results are displayed, select the entry for Homo sapiens. The primary accession number associated with human CYP2C9 is P11712. The entry page for this accession number offers a detailed summary of the protein. It describes CYP2C9 as a cytochrome P450 monooxygenase that plays a role in the metabolism of many endogenous substances, such as fatty acids, as well as a wide array of clinical drugs. Specifically, it mentions drugs like S-warfarin, phenytoin, and diclofenac. Additionally, the entry confirms that the protein is located within the membrane of the endoplasmic reticulum. This subcellular localization is essential for its function in the metabolism of drugs in the liver. To investigate variants, navigate to the section labeled Disease and Variants on the left-hand menu. This section contains a table listing known natural variants. To find a specific variant, you can search or scroll through the table until you locate the one corresponding to the reference single nucleotide polymorphism, RS1057910. The UniProt database will display this variant by its amino acid change, specifically P.Al359Lu, which is also known as I359L. UniProt provides a detailed description of the variant's impact. It connects this change to the CYP2C9 gene variant known as the CYP2C9*3 allele. The description notes that this variant is associated with impaired metabolism of multiple substrates and contributes to the poor metabolizer phenotype for certain drugs, such as the antiepileptic drug phenytoin. Additionally, UniProt offers links that direct users back to the original database entry for the variant, as well as to scientific literature that describes the findings related to this variant. This process demonstrates how UniProt serves as a crucial bridge, translating the DNA-level information from DBSMP into a protein-centric context and providing expertly curated summaries of a variant's known functional effects. The Protein Data Bank, PDB, is the single global archive for experimentally determined three-dimensional, 3D, structures of biological macromolecules. Visualizing the protein structure allows for a mechanistic understanding of how a variant might alter its function. In this case, we must use the PDB to visualize the 3D structure of CYP2C9, locate the position of the I359L substitution, and hypothesize how this change impairs enzyme function. The process begins by searching the Protein Data Bank, a comprehensive database of protein structures. To do this, navigate to the homepage of the Research Collaboratory for Structural Bioinformatics Protein Data Bank and search for the term CYP2C9. This search will yield multiple structures related to this protein. For our analysis, it is important to identify two key types of structures. The first type comprises wild-type structures, which are the arrangements of the normal, non-variant form of the protein. These structures are often available in complex with a substrate or an inhibitor, and examples include the structure with the identifier 1 open parenthesis, 10 capital letter G, 5, close parenthesis, the structure with the identifier 4N, Z, 2, the structure with the identifier 5W, 0, C, among others. These structures serve as a baseline, providing insights into the protein's normal conformation. The second type involves the structure of a variant form of the protein. Of particular interest is the structure corresponding to the CYP2C9 enzyme, with a specific mutation known as the variant. This structure has been determined and is available in the Protein Data Bank with the identifier 5X24. The availability of this structure allows for a direct comparison between the normal and variant forms of the protein. The built-in mole * molecular viewer on the Protein Data Bank website can be used to analyze entry 5x24. To begin, locate residue 359, which is a leucine in this structure, using the sequence viewer. Observe its position within the overall fold of the protein. It becomes clear that this residue is not part of the central active site that contains the heme group where catalysis takes place. At first glance, one might think that a mutation occurring far from the active site would be harmless. However, the publication associated with the structure 5x24 reveals the true mechanism behind its effects. The substitution of isoleucine with leucine at position 359, despite being distant from the active site, remarkably causes changes in the side chains of residues near the active site and the access channel. This long-range conformational change disrupts the precise geometry necessary for optimal substrate binding and enzymatic activity. This structural analysis reveals a sophisticated principle of protein biology. A variant's functional impact is not solely determined by its proximity to the catalytic center. The I359L substitution in CYP2C9*3 is a classic example of how a seemingly conservative amino acid change can induce allosteric or long-range conformational shifts that propagate through the protein structure. These shifts can alter the shape of the substrate binding pocket or the channel through which the substrate enters, thereby impairing enzyme function. This understanding moves beyond a simplistic lock-and-key model of enzyme function to a more dynamic and interconnected view of protein structure, which is essential for accurately interpreting the impact of many pharmacogenomic variants. The ultimate goal of interpreting a pharmacogenomic variant is to predict its clinical consequence, or phenotype. This involves translating an individual's diplotype, their combination of two-star alleles, into a clinically meaningful category that can guide drug selection and dosing. This translation is most established for proteins involved in drug absorption, distribution, metabolism, and excretion, ADME, particularly drug-metabolizing enzymes and drug transporters. Genetic variations in drug-metabolizing enzymes are the most well-characterized cause of interindividual differences in drug response. These variations lead to distinct metabolizer phenotypes that directly influence drug exposure and, consequently, safety and efficacy. Based on their genetic makeup, individuals can be classified into distinct metabolizer groups. This classification is often formalized using an activity score system, where each allele is assigned a value reflecting its function: 1.0 for a fully functional allele, 0.5 for a reduced function allele, and 0 for a non-functional allele. An individual's diplotype activity score is calculated by summing the scores for their two alleles. This score helps predict their metabolic phenotype. Individuals classified as poor metabolizers possess two non-functional alleles, resulting in an activity score of 0. They have little to no enzyme activity. Intermediate metabolizers carry either one reduced function allele and one non-functional allele, or two reduced function alleles, leading to an activity score of either 0.5 or 1.0, and thus have decreased enzyme activity. Normal metabolizers have two fully functional alleles, which corresponds to an activity score of 2.0. They serve as the standard reference for drug metabolism. It is worth noting that some classification schemes group individuals with an activity score of 1.5, such as those with one normal and one reduced function allele, as normal metabolizers. Ultra-rapid metabolizers possess more than two copies of functional alleles due to gene duplication copy number variations, resulting in an activity score greater than 2.0. These individuals exhibit significantly increased enzyme activity. The gene CYP2D6 is a classic example of a highly polymorphic gene that illustrates how different types of genetic variation lead to these distinct phenotypes. It is responsible for the metabolism of approximately 20% of commonly prescribed drugs, including many antidepressants, antipsychotics, beta blockers, and opioids. The phenotype known as poor metabolizers is often caused by carrying two copies of non-functional alleles, such as CYP2D6*4, which contains a splice site mutation leading to an inactive protein, or CYP2D6*5, which represents a whole gene deletion. The intermediate metabolizer phenotype can result from various combinations of alleles, such as one copy of the CYP2D6*4 allele, which has no function, and one copy of the CYP2D6*41 allele, which has reduced function. The ultra-rapid metabolizer phenotype is caused by copy number variations where an individual inherits duplications or multiplications of a functional allele. Examples include duplications of the CYP2D6*1 or CYP2D6*2 alleles, which lead to enzyme activity levels that are higher than normal. The clinical consequence of a patient's metabolizer status is not inherently good or bad. It is entirely dependent on the pharmacology of the specific drug being administered. This creates a fundamental dichotomy between active drugs and prodrugs that is the cornerstone of clinical pharmacogenomic interpretation. Active drugs, such as tricyclic antidepressants, are compounds that become active upon administration and are later inactivated through the process of metabolism. In individuals with a poor metabolizer profile, the drug is eliminated from the body slowly, which can lead to accumulation. This buildup results in elevated concentrations of the drug in the plasma and increases the risk of dose-dependent toxicity. Conversely, in individuals with an ultra-rapid metabolizer profile, the drug is cleared from the body too quickly. This rapid clearance often results in the drug concentrations in the blood being too low to be effective, which can lead to treatment failure. Prodrugs such as codeine and tamoxifen are inactive when administered and must be metabolically converted into their active forms. Codeine, an opioid pain reliever, is transformed into its active metabolite, morphine, primarily through the action of the enzyme CYP2D6. When individuals have a poor metabolizer profile for this enzyme, the conversion is minimal or absent, resulting in a lack of effective pain relief. Conversely, individuals who are ultra-rapid metabolizers produce high levels of morphine quickly, which can be potentially toxic. This rapid conversion increases the risk of severe respiratory depression and overdose even from standard doses of codeine. Tamoxifen is used in the treatment of breast cancer and is converted by the enzyme CYP2D6 into its highly potent active metabolite, endoxifen. Poor metabolizers of this enzyme produce very little endoxifen, which has been linked to a higher risk of breast cancer recurrence and poorer clinical outcomes. The enzyme UDP-glucuronidyltransferase 1A1, encoded by the UGT1A1 gene, is a phase II metabolizing enzyme responsible for conjugating and activating various compounds, including bilirubin and SN38, the active metabolite of the chemotherapy drug irinotecan. The most well-known variant is UGT1A1*28, which involves a variation in the number of TA repeats in the gene's promoter region. The presence of 7 repeats instead of the normal 6 reduces gene transcription, leading to decreased UGT1A1 enzyme levels. Individuals who are homozygous for the 28 allele, 28/*28, have significantly impaired ability to inactivate SN38. This leads to accumulation of the toxic metabolite, placing them at a high risk for severe, life-threatening neutropenia and diarrhea when treated with standard doses of irinotecan. Genotyping for UGT1A1*28 is now commonly used to guide irinotecan dosing. While metabolizing enzymes alter the chemical structure of drugs, drug transporters function as gatekeepers, regulating their movement across biological membranes. They are essential for drug absorption in the intestine, uptake into the liver for metabolism, excretion by the kidneys, and crossing the blood-brain barrier. Genetic variation in these transporters can have clinical effects as significant as those caused by metabolic enzymes. A notable example is the solute carrier organic anion transporter family member 1B1, commonly known as OATP1B1, encoded by the SLCO1B1 gene. This protein is expressed on the sinusoidal, blood-facing, membrane of liver cells and is responsible for actively transporting many drugs from the bloodstream into the liver, including the widely prescribed HMG-CoA reductase inhibitors, statins. Several common SNPs in the SLCO1B1 gene impair the transport function of the OATP1B1 protein. The most studied among these is C.521T greater than C, p.val174ala, which defines haplotypes like SLCO1B1*5 and SLCO1B1*15. Individuals with 1 or 2 copies of the C allele experience significantly decreased hepatic uptake of statins such as simvastatin and atorvastatin. This reduction means less drug reaches the liver, the site of action and metabolism, and as a result, more drug stays in the systemic circulation. The higher plasma concentrations substantially raise the risk of the primary dose-dependent toxicity of statins, myopathy, which can range from muscle pain to life-threatening rhabdomyolysis. The clinical scenario caused by a defect in an uptake transporter like SLCO1B1 effectively mimics that of a metabolic defect. In both situations, either the drug cannot enter the liver efficiently, transporter defect, or it cannot be broken down properly once inside, metabolic enzyme defect. The result is increased systemic drug exposure and a higher risk of toxicity. This demonstrates that transporters are often the limiting step in the disposition of many drugs. Their genetic variation is therefore just as important clinically as that of metabolizing enzymes, emphasizing the need for a comprehensive view of ADME genetics that goes beyond a singular focus on the CYP450 system. While pharmacogenomics has traditionally focused on optimizing the use of small molecule drugs, its importance has grown even more in the age of advanced therapies, where it is often essential. For many biologics, such as monoclonal antibodies, and cell and gene therapies, CGT, genetic or molecular testing is not just a tool for dose optimization, but a crucial step in determining patient eligibility. This testing ensures that these highly specific and often expensive therapies are given only to patients who can benefit from them or who will not be unduly harmed. The use of companion diagnostics for biologic therapies can be understood through 3 fundamental strategic pillars: ensuring safety, identifying the drug's target, and assessing for primary resistance mechanisms. The human leukocyte antigen, HLA, system plays a crucial role in the immune system's ability to differentiate between self and non-self. Among its components, the HLA-B*5701 allele is significantly associated with a severe and potentially fatal multi-organ hypersensitivity reaction, HSR, to the antiretroviral drug abacavir. This hypersensitivity mechanism involves the presentation of the drug by the HLA-B*5701 protein, which triggers a robust T-cell response. The clinical utility of testing for this marker is particularly high due to its near 100% negative predictive value. In other words, a patient who tests negative for HLA-B*5701 has virtually no risk of developing an immunologically mediated hypersensitivity reaction. As a result, Screening for the HLA-B*5701 allele has become the standard of care worldwide before starting abacavir therapy. If a patient's test result is positive, indicating the presence of HLA-B*5701, the use of abacavir is strictly contraindicated. This critical information is permanently recorded in the patient's medical record as a drug allergy, ensuring that it is considered in future treatment decisions. Trastuzumab, Herceptin, is a humanized monoclonal antibody designed to target the human epidermal growth factor receptor 2, HER2, a receptor tyrosine kinase that promotes cell growth and proliferation. In about 15 to 20% of breast cancers and some gastric cancers, the ERBB2 gene, which encodes HER2, is amplified, causing significant overexpression of the HER2 protein on the tumor cell surface. This overexpression is a key factor in tumor growth. Trastuzumab works only in these HER2-positive tumors, as it needs the presence of its target to be effective. Therefore, testing for HER2 status is mandatory before starting therapy. This is usually done with immunohistochemistry, IHC, to evaluate protein overexpression or fluorescence in situ hybridization, FISH, to assess gene amplification. Only patients with tumors confirmed to be HER2 positive are eligible for and will benefit from trastuzumab and other anti-HER2 targeted treatments. Cetuximab and panitumumab are monoclonal antibodies that target and block the epidermal growth factor receptor, EGFR, another receptor tyrosine kinase involved in cell signaling. The EGFR signaling pathway proceeds through a downstream cascade that includes the key signaling protein KRAS. In about 40% of metastatic colorectal cancers, the KRAS gene has an activating somatic mutation. This mutation keeps the KRAS protein in a permanently on state, causing the signaling pathway to be constantly active regardless of the upstream EGFR status. Consequently, blocking EGFR with cetuximab or panitumumab is ineffective in these patients. The tumor's growth signal bypasses the blockade. This makes the presence of a KRAS mutation a strong negative predictive biomarker. Testing for somatic KRAS mutations in tumor tissue is now standard practice for all patients with metastatic colorectal cancer being considered for anti-EGFR therapy. Patients with tumors harboring a KRAS mutation are not candidates for this class of drugs, as they are known not to respond. These three landmark cases are more than isolated examples. They embody the core strategic principles of companion diagnostics for modern biologic therapies. They illustrate how molecular testing is applied for 1. Safety screening, identifying patients at high risk of severe adverse events, such as with HLA-B*5701. 2. Target identification, identifying the specific patient population whose disease expresses the drug's molecular target, such as HER2. And 3. Resistance assessment, excluding patients whose disease carries mutations in downstream pathways that cause primary resistance, like KRAS. This triad offers a solid framework for understanding the critical connection between a specific biologic drug and its essential biomarker test. For the most advanced therapeutic modalities, cell and gene therapies, the role of genetic testing is even more fundamental, though its purpose differs significantly between therapy types. The term genetic testing, in the context of CGT eligibility, can refer to two distinct processes: diagnosing an inherited disease or characterizing a tumor's molecular profile. Gene replacement therapies are designed to treat monogenic diseases by delivering a functional copy of the gene that is defective in the patient. The therapeutic principle is to correct the root cause of the disease at the DNA level. Consequently, eligibility for these therapies is entirely dependent on a definitive genetic diagnosis that confirms the presence of pathogenic mutations in the specific target gene. Zolgensma, onisimnogene abeparvovec, is an approved gene therapy for spinal muscular atrophy, SMA. Its use is limited to patients with a confirmed diagnosis of SMA caused by biallelic, i.e., on both chromosomes, mutations in the SMN1 gene. Luxturna, voretigene nepervovec, is approved for a rare form of inherited retinal dystrophy. It is indicated only for patients with confirmed biallelic mutations in the RPE65 gene. Casgevy, exagamblagene autotemsil, and Lyfgenia, Levodobeglogene autotemsil, are gene therapies for sickle cell disease, which is caused by mutations in the beta-globin (HBB) gene. In these cases, the genetic test is fundamentally diagnostic. It identifies the patient's inherited monogenic defect, which is the direct target that the therapy is designed to correct. Chimeric antigen receptor (CAR) T-cell therapy is a form of immunotherapy, not a gene replacement therapy for an inherited disease. In this process, a patient's own T-cells are collected and genetically engineered ex vivo to express a synthetic receptor, the CAR, on their surface. This CAR is designed to recognize and bind to a specific protein, an antigen, that is present on the surface of the patient's cancer cells. Eligibility for currently approved CAR T-cell therapies is therefore not based on the patient's germline DNA. The determination of appropriate therapy largely depends on two main factors: the type of cancer and antigen expression. First, the patient's cancer must be of a type known to express the target antigen. For instance, therapies such as Kymriah, tisagenlecleucel, and Yescarta, axicabtagene ciloleucel, are approved for B-cell malignancies, including leukemias and lymphomas, because they target the CD19 antigen, which is consistently expressed on B cells. Similarly, Abecma, idacabtagene vicleucel, is used for multiple myeloma and targets the BCMA antigen. To confirm the presence of the target protein, an immunohistochemical or flow cytometry assay is performed on tumor cells. Secondly, clinical history plays a crucial role. These therapies are typically approved for patients with relapsed or refractory disease who have not responded to multiple prior lines of standard treatment. This reveals a crucial distinction often conflated in discussions of advanced therapies. For gene replacement, the prerequisite test is diagnostic and pharmacogenomic, identifying the inherited defect in the patient's genome. For CAR-T therapy, the prerequisite test is molecular pathology, identifying a protein target on the tumor cell, a characteristic of the disease rather than the patient's constitutional DNA. This distinction is vital for understanding the different scientific and clinical rationales underpinning testing requirements in the rapidly evolving CGT landscape. The journey of a pharmacogenomic marker from a research discovery to a routine clinical test is a complex, multi-stage process that extends far beyond the initial scientific validation. For a test to be successfully integrated into healthcare, it must navigate a translational gauntlet that includes demonstrating rigorous analytical and clinical performance, proving tangible benefit to patients, gaining regulatory acceptance, and addressing significant ethical, legal, and social challenges. This comprehensive evaluation ensures that new tests are not only accurate, but also safe, effective, and equitably implemented. The ACC model, developed with support from the Centers for Disease Control and Prevention (CDC), offers a systematic and evidence-based framework for evaluating genetic tests. Its name is an acronym representing its four core components, each of which must undergo thorough assessment. The first component is analytical validity, which focuses on the performance of the laboratory assay itself. It questions how accurately and reliably the test measures the genetic marker of interest. Key metrics in this area include analytical sensitivity, the ability of the test to correctly identify individuals with the variant; analytical specificity, its ability to correctly identify those without the variant; and reproducibility, the consistency of results across different operators, laboratories, and time. The second component is clinical validity. This evaluates the strength and reliability of the association between the genetic marker and a clinical outcome or phenotype. It asks how consistently and accurately the genotype predicts the phenotype. This involves examining factors such as clinical sensitivity, clinical specificity, and the positive and negative predictive values of the test within a specific population. For example, the clinical validity of HLA-B*5701 testing for abacavir hypersensitivity reaction is extremely high. In contrast, the connection between some CYP2, C19 variants and clopidogrel outcomes has shown more variability across different studies. The third component, clinical utility, is often the most challenging to establish, but is crucial. It moves beyond mere prediction to question whether using the test and acting on its results actually lead to improved health outcomes for patients. Demonstrating clinical utility typically requires evidence from large, prospective, randomized controlled trials. For instance, such studies may show that genotype-guided dosing of a drug reduces adverse events or enhances efficacy compared to empirical dosing. A lack of robust clinical utility data remains a significant barrier to the widespread adoption and reimbursement of many pharmacogenomic tests. Finally, ethical, legal, and social implications, ELSI, must be considered. This component involves thoroughly contemplating the potential societal consequences of implementing genetic testing. Addressing these issues proactively is essential to ensure the responsible deployment of this technology. The integration of pharmacogenomic testing into routine care raises significant ELSI challenges that must be addressed to foster public trust and ensure equitable use. As detailed in the ACC framework, these issues are not peripheral but are a core component of a test's overall evaluation. Key concerns related to genetic information include issues of discrimination, privacy, and informed consent. A primary worry among patients is that genetic data revealing a predisposition to certain diseases or adverse drug reactions could be exploited by employers or insurance companies to discriminate against them. For instance, individuals might face denial of employment or health coverage based on their genetic profile. While legislation such as the Genetic Information Non-Discrimination Act in the United States provides some protections, gaps remain, and public awareness about these issues is still limited. Privacy and confidentiality are also major concerns. Genetic data is uniquely personal and permanent, making it essential to have robust systems for data security and clear policies governing access to and sharing of this information. As electronic health records and large-scale data repositories become more commonplace, safeguarding patient privacy becomes increasingly complex. Furthermore, obtaining informed consent for pharmacogenomic testing involves a complex process. It must thoroughly explain not only the purpose of the testing but also its limitations, including the possibility of uncertain or incidental findings. These are discoveries that reveal risks for conditions unrelated to the original test. Additionally, patients should be informed about how their data will be stored and used in the future. A strong, biologically plausible association between a gene variant and a drug response is merely the first step on a long and arduous path to clinical implementation. For a pharmacogenomic test to be widely adopted, it must successfully pass through this translational gauntlet. This multi-stage process, which demands rigorous proof of analytical validity, and clinical utility, alongside regulatory approval, pharmacoeconomic justification, and the resolution of complex ethical and social issues, explains the significant lag time often observed between a scientific discovery and its routine use at the patient's bedside. Regulatory agencies like the U.S. Food and Drug Administration, FDA, and the European Medicines Agency, EMA, play a pivotal role in the implementation of pharmacogenomics. Their oversight ensures that pharmacogenomic information used in drug development and clinical practice is based on sound scientific evidence. The key regulatory activities involve several important processes. Firstly, agencies such as the Food and Drug Administration and others issue guidance documents for the pharmaceutical industry. These documents provide instructions on how and when to integrate pharmacogenomic studies into drug development programs, covering stages from preclinical research to phase 3 clinical trials. Secondly, the regulatory bodies review genomic data through initiatives like the Voluntary Exploratory Data Submissions Program. This program encourages pharmaceutical companies to share exploratory genomic data in a non-binding manner. The goal is to facilitate scientific exchange and help regulators and sponsors understand the potential regulatory implications of new biomarkers early in the development process. Finally, one of the most significant areas of regulatory impact is drug labeling. Depending on the strength of the evidence, a drug's label may include pharmacogenomic information. This information can range from cautionary statements to recommendations for genetic testing. In some cases, it may even require testing with an FDA-approved companion diagnostic before the drug is prescribed, as seen with medications such as trastuzumab and abacavir. The field of applied pharmacogenomics is at a pivotal point, shifting from a research-focused area to a vital part of modern clinical practice. Moving from discovering a genetic variant to developing a test that guides treatment exemplifies the integration of molecular genetics, clinical pharmacology, bioinformatics, and regulatory science. As this report highlights, understanding the functional effects of genetic markers, from SNPs that modify enzyme activity to gene amplifications that determine eligibility for targeted biologics, is central to the goal of precision medicine. Analyzing key pharmacogenomic markers such as variants in CYP2D6, UGT1A1, and SLCO1B1 shows a clear, predictable link between genotype and drug behavior, offering practical strategies to reduce toxicity and improve effectiveness for an increasing number of small molecule drugs. Using bioinformatics tools like DBSNP, UniProt, and the PDEB has become essential, allowing practitioners to explore the effects of variants from population patterns down to their atomic structural impacts. Additionally, molecular testing has evolved from a support tool to an essential requirement for many advanced therapies. For biologics like trastuzumab and abacavir, as well as innovative cell and gene therapies, a patient's molecular or genetic profile is the key factor in determining eligibility. This shift emphasizes that the future of therapeutics is deeply connected to diagnostics. Nevertheless, widespread clinical adoption faces obstacles beyond scientific research. Successfully implementing a pharmacogenomic test depends on navigating the translational gauntlet, a rigorous evaluation process outlined by the ACC framework. Demonstrating not just analytical and clinical validity, but also real clinical utility is crucial. This must happen within a strong regulatory system and a healthcare infrastructure capable of addressing ethical, legal, and economic challenges. As sequencing costs decline and our understanding of the genome advances, the main barrier to realizing the potential of pharmacogenomics will not be data generation, but the multidisciplinary effort needed to translate that data into evidence-based, equitable, and effective patient care. This was an episode of the Nexus Clinical Podcast. This is for general informational purposes only and does not constitute the practice of medicine, nursing, or other professional healthcare services, including the giving of medical advice. No doctor-patient or pharmacist-patient relationship is formed. Using this information and the materials linked to this content is at the user's risk. This is not intended to substitute for professional medical advice, diagnosis, or treatment. Users should not disregard or delay seeking medical advice for any medical condition they have, and they should consult their healthcare professionals for any such conditions. Clinical experts created the references, content, and clinical insight. NotebookLM, a Google AI tool, created the content, which I extensively reviewed before release. Finally, the host states that he takes all conflicts of interest seriously. Currently, there are no conflicts to disclose. For all of his disclosures and the companies he invests in or advises, he directs users to reach out independently, where he keeps an up-to-date and active list of all disclosures.